Welcome and greeting to all. My name is Dr. Irfan Ahmed and I work as a medical lecturer in Microbiology, Virology and Immunology Department of the Samarkand State Medical University. Today we will going to discuss on the significance of the microbiology in practical activity of the doctor, the history of the microbiology, classification and the structure of the microorganism. So let's move forward. This lecture schedule will divide it into three components: his history of microbiology, classification of the bacteria, structure of the bacterial cell. So microbiology is a science which study the most shallow living creatures, microorganisms. Before the invention of the microscope, humanity was in dark about their existence. But during the centuries, people could make us of process the vital activity of the microbe for its need. They could prepare a cumin, alcohol, wine, vinegar, bread, and other products. During many centuries, the nature of the fermentation remained incomprehensible. So this picture is describe you the Bruegel, the triumph of the death, 1560. Microbiology learns the morphology, physiology, genetics, and the microorganism systematization, their ecology, and the other life form. So, specific class of the microorganism: algae, protozoa, fungi, yeasts, and molds. Bacteria, rickets, viruses, prions. Microorganisms are the extraordinary, widely spread in the nature. They literally ubiquitous forward us from birth to our death. Daily, hourly, we eat up thousands and thousands of the microbes together with the air, water, food, on our skin, in mouth and nasal cavities, on mucous membrane and in bowels. In numerous amount of the microorganisms live and act. Many of them are found in earth, cortex, and in the air and in the ocean, sea, rivers, water, on all of the latitude, mineral lands, and continents. For the first time, term the microbe was offered by the French scientist H.S. Satilot in 1878. It derived from the Greek microbe, that means briefly living or the most shallow living creatures. Science which learned the microorganism was named by the Douglas microbiology. For shortly development period of this science accumulated great factual material. The separate microbiological branches such as bacteriology, mycology, parasitology, proteology, virology, and quick appeared. This is a comprehensive size of the bacteria. You can notice on the Staphylococcus, rhinovirus, Ebola virus, rugweed, pollen, lymphocyte, yeast, etc. Period of the microbiology development: morphologic, physiologic, and prophyletic. Development of the microbiological science was interlinked with the art of glass and diamonds grinding. This brought to creation of the first microscope by the Hans and Zakarian Jens in Holland in 1590. The discovery of the microorganism is associated with the name of Anthony Van and Leeuwenhoek. In 1683, Leeuwenhoek describes the basic bacterium form. His scientific supervision, Leeuwenhoek described in the special letters and the sent of them to the London Royal Scientific Society. He sent away about 300 letters. The Leeuwenhoek letters brought on the enormous surprise among the English scientists. They opened a fantastic world of the invisible creatures. He named them living animals, anima lacula viva. And one of later wrote, in my mouth there are more animalcula viva than people in all United Kingdom. These wonderful discovery of the Dutch naturalist were the embryo which science of the bacteria developed, namely from the time of start so-called is morphological period in microbiology. History, the 17 middle of age. It is the also called the morphological period as the study of the microorganism come only to describe of their dimension and forms, biological properties and their significances for all men still a long time remained incomprehensible. However, using the primitive microscope of that time, it was difficult to determine the difference between the separate bacteria species. Even celebrated founder of the scientific systematization of all of living organisms, Carl Lenny renounced to classify the bacteria. He gave them general name cows. Edward Jenner vaccinated James Phipps with cowpox material, resulting in the development of the resistance to the smallpox infections to the boy and thereby the establishing the scientific credibility of the vaccination to prevent the disease. In the second half of the 19th century, microbiology strongly affirms an independent science, namely these sciences were fruitful soil on which Pasteur's talent evinced. He studied wine, illness, fermentation, made the pasteurization method offered to the grow microbes on the artificial nutrients media. He proved that on the definite cultivation conditions, the pathogenic bacterial loss its virulence, made vaccine against the anthrax and rabies. In picture, Pasteur's administration rabies to the Josepher Masters, 9 years old boy who had been bitten by a rabbit dog, which vaccine saved the boy's life. Not less important are the scientific work of the celebrated German scientist R. Koch. He performed classic research on the etiological of the anthrax, open tuberculosis bacilli, Vibrio cholera, and proposed to the isolate the pure bacterial culture on the solid nutrient media, which is gelatin and potatoes. The development to the preparation staining method by the alanine dyestuffs method of the hanging drop of examination of the bacterium motility offered apparatus for the sterilization. The patriarch of the world and Ukrainian microbiology met Chinikov. He studied inflammation pathology, phagocytosis based about the antagonism of the bacteria from all microbes antagonist. The Metichinikov preferred the lactic bacteria on the basis of offered three medical preparations: sour, clotted milk, yogurt, and lactobacilline. Now they are called by the eubiotics. Classic Metchnikoff research defined as a prophylactic period in the microbiology history. In 1892, the D. Ivanovisky described a virus of the mosaic tobacco illness, new class of the infectious agent. Microorganisms constitute a very antiquity group of the living organism which appeared on the earth's surface almost the 3000 million years ago. There are natural and artificial classification system. Berge Manual of the Determinative Bacteriology, the Bible of the bacterial taxonomy. So now classifying the bacteria. Berge Manual of the Systemic Bacteriology describes the classify the bacteria via the evolutionary or the genetic relationship. The second one is Berge Manual of the Determinative Bacteriology, classifies the bacteria by the cell wall composition, morphology, biochemical test, differential staining, etc. Now you can see the five kingdom systems of the classification, the three domain systems, comparison of the three domains like eubacteria, archaea and eukarya by the characteristics. So first we will describe the cell type. Prokaryotic, eubacteria have the prokaryotic, archaea as a prokaryotic and eukarya is a eukaryotic. So cell wall is a peptidoglycan, varies, membrane lipids, eubacteria has a unbranched, archaea has a branched, eukarya has a unbranched. Sensitive to the antibiotic, which is eukaryotic is a yes, is a sensitive. Archaea is a no sensitive, eukarya is a no sensitive. Circular chromosomes, eubacteria has a yes, archaebacteria has a yes, and eukarya is no except in the mitochondria and the chloroplast. Histones, eubacteria have no histones, archaea and eukarya have the histones. The prokaryotes, archaea and bacteria, universal ancestors. So you can see the cyanobacterium, different shapes and types of the bacteria which is following in your screen. So spirochetes, gram negative bacteria, rickettsia, archaeobacteria, gram positive bacteria, mycoplasma. So this picture you can also notice the eukarya and bacteria. This is size, shapes and different family, orders, classes, phylum and genus and species have been discussed already. So classification system in the prokaryote. The first one is microscope morphology, microscopic morphology, colony appearances, physiological, biochemical characteristics, chemical analysis, serology analysis, genetic and molecular analysis, which have been the following is G plus C base composition, DNA analysis using genetic probes, nucleic acid sequence and rRNA analysis. The bacterial taxonomy based on the Berge materials. The Berge manuals of determinative bacteriology, 5-volume resources conveying all know the prokaryotes. The first one is classification based on the genetic information, phylogenetic. The two domains, archaea and bacteria, 5 major subgroups with 25 phyla. So next is the taxonomy. The domain is kingdom, phylum, class, order, family, genus and species. The next is major taxonomical group of the bacteria. Volume 1A domain archaea, which is primitive, adapted to extreme habitat and the mode of the nutrition. Volume 1B domain bacteria. Volume 2 to 5 is phylum Proteobacteria, which is gram-negative cell walls. Phylum Firmicutes, mainly gram-positive with low G plus C content. Phylum Actinobacteria, gram-positive with the high G and C content. The next one is microbiological phylogeny. The phylogeny of the domain bacteria, which is the second edition of the Berge Manual of the Systemic Bacteriology, divided into the domain bacteria into the 23 phyla. So microbial phylogeny is a phylogeny of domain bacteria. The phylum Proteobacteria, the largest group of the gram-negative bacteria, extremely complex group with our 400 genera and the 1300 named species. All major nutritional types are represented: photography, heterotrophy, and several types of the chemolithotropy. Next is a sometime called the purple bacteria, although very few are the purple. The term referred to a hypothetical purple photosynthetic bacterium from which the group is believed to have the evolved. The slide is continue. The phylum Proteobacteria continued, divided into the 5 class: alpha Proteobacteria, beta Proteobacteria, gamma Proteobacteria, delta Proteobacteria, and epsilon Proteobacteria. This slide is also continue. The family Enterobacteriae, the gram-negative enteric bacteria, which include the E. coli, Proteus, Enterobacter, Klebsiella, Salmonella, Shigella, Serratia, and others. The family Pseudo, which include the genus Pseudomonas and related the genera. Other medically important proteobacteria include the genera Haemophilus, Vibrio, Campylobacter, Helicobacter, Rickettsia, and Brucella. The phylum Firmicutes, low G plus C gram positive bacteria, divided into 3 types. The first class is Clostridia, include the genera Clostridium and Desulfotomaculatum and others. Class 2 is Moticutes, bacteria in the class cannot make the peptidoglycan, and the lac cell wall include the genera Mycoplasma, Ureaplasma, and other. Class 3 Bacilli include the genera Bacillus, Lactobacillus, Streptococcus, Lactobacillus, Geobacillus, Enterobacillus, Listeria, and Staphylococcus, and others. The phylogeny of the domain bacteria: high G plus C gram positive bacteria. The next one is include the genera Actinomyces, Streptomyces, Corynebacterium, Micrococcus, Mycobacterium, and Propionibacterium acne. The phylum Chlamydia, small phylum containing the genus Chlamydia. Phylogeny of the domain bacteria: the phylum Spirochetes. The spirochetes is characterized by the flexible helical cells with a modified other outer membrane, the outer sheet, and modified the flagella, axilla, filaments located within the outer sheet. Important pathogenic genera include the Treponema borrelia and Treponema pallidum. The phylum Bacteroides include the genera Bacteroides, Flavobacterium, the Flexibacterium, and Flexibacterium, Cytophylogenia, Flexibacteria, the Cytophagia, and motile by the means and grinding motility. Prokaryotes kingdom has 4 divisions according to the structure of the cell wall and gram staining. The gracilicutes, gracilous thin cutaneous skin. Gram negative bacteria: Firmicute, Firmus, Firm. Gram positive bacteria: Tannericutes, Tanner, soft tanner, microbe within the cell wall. Mendosicuites, Mendos, mysticate, microbe with antipical peptoglycan. Bacterial nomenclature: binomial naming system, 2 words naming systems. The first word is genus name, always capitalized, means Escherichia. And the second word is a species name, not capitalized, coli. When writing full name, genus, usually abbreviated E. coli. Full name always italicized which underlined. Species is a population of the microorganism which have the only source of origin common genotypes and during the present stage of the evolution are characterized by the similar morphological, biochemical, physiological and other signs. If deviation from the typical species properties and are found on the examination of the isolated bacteria, then culture is considered a subspecies. Infraspecies subdivisions serover, morphover, which is morphological properties, chemophover is a chemical properties, biover, biochemical or physiological properties, pathover, pathological properties, phagover is isolation and relation to the phagis. The term clone was applied to the population of the cell derived from a single cell. Population is an elementary evolution unit structure of the definite species. The term strain design a microbiological culture obtained from the different source or from the one source but at different times or a subgroup within a species with one or more characteristics that distinguish it from the other subgroups in the species. Bacteria are the unicellular organism lacking the chlorophyll too. Morphological classification of the bacteria. Morphological bacteria possess four main forms. Spherical, which is Koki, rod-shaped bacteria, bacilli and Clostridia. The spiral shape, Vibros, spirilla and spirochetes. Threaded shaped, non-pathogenic. You can see the following the pictures with the name of the bacteria like coccus, rod, bacilli, curved form, spirillum, spirochetes, diplococci, Neisseria, Guneria, is a coffee bean shaped, is coco bacilli, vibrocolera, trades, strychnine, mycobacterium, cornybacterium, spirilla, spirochetes, spore forming rods, streptococci is more on. The Koki group, the coccus is a diplococcus, then streptococcus is tetrad, is a sarcini and staphylococcus. The Koki. The Koki form are the spherical ellipsoidal bean shaped and lanceolate. Is the Koki are the subdivided into the 6 groups according to the cell arrangement, the divisions and biological properties. The micrococi, the cells are the arranged singly or the irregularly. They are saprophytes and live in the water and air. Diplococci is a diplos double divided into one plane and remain attached in the pair. These include the meningococcus, the causative agent of the epidemic cerebrospinal meningitis, and the gonococcus cause the agent of the gonorrhea and blinoria. The pneumococcus causative agent of the pneumonia. The streptococcus divided into the one plane and are the arranged in the chain of the different length. Some streptococci are pathogenic for the human and are the responsible for the various disease. The tetracocci divided into the two plane at right angle to one another and form the groups of the four. They very rarely produce a disease in human. Staphylococci, which is a cluster of grapes, divided into the several plan, resulting in the irregular benches of the cells, sometimes resembling cluster of the grapes. Some species of the Staphylococcus causative disease in the man and animal. Sarcinae divided into the three plan at right angle to one another and resemble packets of the 8 to 16 or the more cells. They are frequently found in the air. Virulent species have not been encountered. The rods, the rod-shaped form are subdivided into bacteria, bacilli, clostridia. The bacteria include those microorganisms which are, as a rule, do not produce the spores. Coli, bacillus, and microorganisms responsible for the enteric fever, paratyphoid, dysentery, diphtheria, tuberculosis. Bacilli and Clostridia include the organism the majority of the which produce the spores have bacilli responsible for the anthrax, tetanus, anaerobe infectious. According to their arrangement, cylindrical forms can be subdivided into the three groups: Monobacteria, E. coli, Yersinia pestis, Clostridium tetani, and Clostridium botulinum. Diplococci is a pneumoniae and diplocilli. Streptobacteria, Streptobacilli, Bacillus anthrax, and Haemophilus diphtheria. The spiral-shaped bacteria. Vibronus, Vibrocolera are cells which resemble the comma in appearances. Typical representative of this group are Vibros cholera, the causative agent of the cholera, and aquatic Vibrionus, which are widely distributed into the freshwater reservoir. The spirilla are the coiled form of the bacteria exhibiting twisting with the one or more turns. Only one pathogenic species is known, Spirillum mimeus, which is responsible for the disease in human transmitted through the bite of the rat and other rodents, rat bite fever, Sudoku. The Spirillum minus. Spirochetes is spirilla curves means is a corkscrew spiral shapes. Borrelia, the cells have the large obtuse angle, irregular spirals, and the number of the which varies from the 3 to 10. Pathogenic for a man or a causative agent of the relapsing fever transmitted by the lice. Ticks. Borrelia persica, these stains blue-white with the Romanovsky-Jeman stain. Leptospira are characterized by the very thin cell structure. The Leptospira form the 12 to 18 coils wound close to each other, shaping small primary spirals. The organism have the two paired axial filaments attached to the opposite end, which is called the basal bodies of the cell and directed toward each other. Leptospira. Enterogens, which is a pathogenic for the animals and man, cause leptospirosis. Treponema exhibit the thin flexible cells with 6 to 14 twists. The microorganisms do not appear to have a visible axial filaments or an axial crest when viewed under the microscope. A typical representative is the causative agent of the syphilis, Treponema pallidum. Here is the prokaryotic or the eukaryotic difference you can see on your screen with the clear and cut of the differences. The main I will read for you that there is a ribosomes in 70s in the cytoplasm for the prokaryotic. There are the ribosomes from the 80s in the cytoplasm for the eukaryotic. In prokaryotic, peptidoglycan are included in the cell wall, which is murine. In eukaryotic, peptidoglycan are not included in the cell wall. In eukaryotic, chromosomes is more than one. There is a mitosis, and chromosomes is a one ball of the double twisted DNA threads. Mitosis is absent in prokaryotic. The next one, you can see the structure of the prokaryotics. And next one, we move the prokaryotic cells and eukaryotic organelle comparisons. In prokaryotics, DNA is circular, histone no, ribosome 70S, and growth is binary fusions. And on the other hand, the eukaryotic cell is linear, histone is yes, ribosome is 80S, and growth by the mitosis. Eukaryotic organelles, mitochondria and chloroplast, circular no, 70S, and binary fusions. And later on you can see the cytoplasmic membrane, endoplasmic reticulum, ribosomes, nucleus, nuclear membrane, cytoplasm, mitochondrion. What is the nucleus? The nucleus is a prokaryotic nucleus, can be seen with the light microscope in the stained material. It is a Fulgen positive, indicating the presence of the DNA. Histone-like properties protein have the recently been discovered in the bacteria, and the presumably play a role similar to the kind of the histone and eukaryotic chromatin. The DNA is seen to be a single continuous gained circular molecule with a molecular weight of the approximately 3x with the power 9 10, the 10 power of 9. The unfold uncleared DNA would be about the 1 to 3 millimeter long, compared with the average length of 1 to 2 micrometer for the bacterial cells. Plasmid. The plasmid is a small circular double stranded DNA, free or integrated into the chromosome. The duplicated and passed on to the offspring, not essential to the bacterial growth and metabolism. May encoded antibiotic resistance, tolerance to the toxic material, enzyme and toxins used in the genetic engineering, rarely manipulated and transferred from the cell to cell. There may be the several differential plasmid in one cell, and the number of the each may vary from the 1 and 100s in the cell. The prokaryotic ribosomes. The ribosomes is a combination of RNA and protein, and the site where the protein synthesis is composed a large 50S and small is 30S. Subunits: S savages is a molecular size. The storage granules, the inclusion granules, in which the metachromatic granules, polysaccharide granules, lipid inclusions, sulfur granules, carboxy enzymes, magnetosomes, which is gas vesicles, Volatin granules, which is Corynebacterium diphtheriae, Neisseria straining, Leofler techniques. On the other hand, the cell envelope. Cell envelope basically is a combination of the two things. Is a cell wall, capsules, and cytoplasmic membrane is also essential but is not include. The cytoplasmic membrane is to act as a physical barrier between the cytoplasm and environments, and selectively control the movement of the substances into out of the cell. Semi-permeable cell wall, the rigid layer that protect the fragile cytoplasmic membrane from the rupturing, maintaining the cell shape. C is a capsule or the slime layer. The cell membrane, the bacterial plasma membrane are the composed of the 40% of the phospholipids and 60% of the protein. The phospholipids are the amphoteric molecules with a polar hydrophilic glycerol head attached via an ester bond to the two non-polar hydrophobic fatty acid trials, which naturally form a bilayer in a aqueous environment. Dispersed within the bilayer are the various structure and the enzymatic proteins which carry out the most membrane functions. As a mesosome, the predominantly function of the bacterial membrane are osmotic or the permeable barrier, location of transport systems of the specific solutes, which is nutrients and ions. Third one is energy generating functions involving the respiratory and the photosensitivity, electron transport system, establishment of the proton motive force and the transmembranous ATP synthesize ATPs, the synthesis of the membrane lipids, which is liposaccharides in the gram negative cells, the synthesis of the membranes, the assembly and the secretion of the extra plasmic protein, coordination of the DNA, replication and segregation with the septum formation and the cell division. The chemotaxis, both motile per se and sensing function, location of the specialized enzyme systems. Cell wall. The unique chemical structure distinguish gram positive from the gram negative bacteria and the archaebacterial species. Rigidity of the cell wall is due to the peptidoglycan compound only found in the bacteria, pseudomurine or other sugar protein glycoproteins. And third one is many antimicrobial interfere with the synthesis of the peptidoglycan. Structure of the peptidoglycan. The basic structure of the peptidoglycan is, number one is altering the series of the two subunits is N-acetylglucosamine, N-acetylmuramic acid, joined subunit from the glyco chain. Glycan chain held together by the straining of the 4 amino acid is a tetrapeptide chain, interpeptide bridge. Differences of the cell wall structure in the gram positive and gram negative cells. And the next one is L forms, which is a gram positive and gram negative, in which describe the gram positive have the cell wall is a peptidoglycan, and on the other hand gram negative is outer membrane is a peptidoglycan, which is a cell wall. Glycocalyx. What is capsule? Protect the bacteria from the phagocytic cells. Slime layer enable the attachment and the aggregation of the bacterial cells. Capsule. The most prokaryotic contain some part of the polysaccharide layer outside of the cell wall polymers. Only capsule of the B anthrax consists of the polypeptide polyglutamic acid. Capsule. What is capsule? Capsule is a covalently bound to the cell wall associated with the virulence in bacteria. And next one example is the Streptococcus pneumoniae. Slime layer. Slime layer is a loosely bonded to the cell. Carbohydrate rich material enhance the adherence of the cells on the surface. Example is the Streptococcus mutans and plaque formation. The next one is biofilms. The slime layer is associated with the aggregation and the formation of the biofilms. Example, Staphylococcus epidermidis biofilms on a catheter tap. The general capsule function is adhesions, avoidance of the immune response, protection from the dehydration, protection of the bacterial cell from the engulfment by the protozoa or white blood cells. The phagocytes are from the attack by the antimicrobial agents of the plant or the animal origin. They provide virulent properties of the bacteria as S. pneumoniae and B. anthracis. Flagella. Flagella have the three parts. Filaments, long, thin, helical structure composed of the protein. Hook curved, sheet, basal body, stack of the rings firmly anchored in cell wall. rotate is a 360 degree. 1 to 2 or many distributed over the entire cells function in the motility. Flagellar arrangements. The monotrichous single flagellum at the one end, which is Cholera vibrio, blue pus bacillus. Lophotrichous small branches of arising from the one to end of the cells, blue green milk bacillus. Alcaligenis facialis. Amphitrichous flagella at the both end of the cells, Spiralium volitans. The fourth one is Peritrichous flagella dispresses over the surface of the E. coli, slowest E. coli, Salmonella of the enteric fever, and paratyphoids A and B. Let's come to the bacterial motility. Flagella are the important for the motility, dispersal, antigenic determinant. Number of the location species is a specific. The rotation of the flagella enable the bacteria to the motile. The pili and fibrae, the short hair-like structure of the surface of the prokaryotic cells, which is protein acious filament, which is a 20 nanometer in diameter. Very common in the gram-negative bacteria, function adherence up to the surface of the substrate, teeth, tissues, involved in the transfer of the genetic information between the cells, having nothing to do with the bacterial environment except the twitching movement of the pseudomonas. You can see in the both surface fibrae are the smaller than flagella and are important too for the attachment. Bacterial endospores. The bacterial endospores are often called the endospore since they are formed within the vegetative cells. The next one is produced in response to the nutrient limitation or extreme environment. The third one is highly resistant. The fourth one is highly dehydrated, means less than 15% water, metabolically inactive, stable for years, not reproductive, function to survive under an extreme growth conditions such as a high temperature, drought, etc. Which example is a Bacillus clostridium, Sporolactobacillus, Thermoactinomyces, Spore Sarcina, Disulfomatomaculum species, Sporulate. So next, which is this is a specific picture of the spore. This is also the spore pictures. The key component, the key composition is dipicolonic acid, calcium structure. The structure is the core cytoplasm, the plasma membrane, core wall, spore wall, cortex, spore coat, exosporium. The next one is endospore. This is the life cycle like a endospore, vegetative cycle, and a sporulation cycle. You can also see the different stages for the spore. The sporulation process begin when the nutritional condition become the unfavorable. The depletion of the nitrogen and carbon source are the both being with the most significant factor. This sporulation involves the production of the many new structures, enzyme and metabolites along with the disappearance of the many vegetative cells component. Spores are the located in the centrally beta anthrax, terminally in the Clostridium tetani, and subterminally in the Clostridium botulinum and Clostridium perfringens, which is very common in formation of pus. The next one is spore of the certain bacilli are the capable of the wide standing boiling and the high concentration of the disinfectant. They are killed in an autoclave exposed to the saturated steam at the temperature of the 115 to the 125 centigrade, and also the temperature of the 150 to 170 centigrade in the pasture hot air oven. So important points. So this lecture is having the no troubles. This is very concise, and try to read text before attending the lectures, and take the time to read as well, because this is extremely important as parallelly in the microbiology. And we have discussed already in the many slides which we discussed in particularly PPT. So good luck for everyone.